on behalf of the selection committee, I'd really like to congratulate Fred. But I wouldn't like to congratulate him because of all these titles, because these titles actually don't mean very much to us on the selection committee. We really look for accomplishment. So somebody who's not accomplished, who has a lot of titles, we could care less. Fortunately, Fred actually has a lot of titles, and he has a lot of accomplishment. So that made our job pretty easy. But the thing that made it the most easy is what I call the 4 Fs. So I'm going to give the F talk now, but not the F-bomb talk, the F talk. And that has to do with Fred, and Fred's not one of the Fs. So the first F that I'd like to say about Fred is that not only is he an accomplished scientist, but he's also a friend to many of us. A friend for many, many years in my case, and certainly an example of how science should be done. So Fred, you're a friend. Now, being a friend is not enough. I have a lot of friends. Well, I have a few friends. Maybe I only have a couple friends. But those friends are not going to get the prize, probably. So what did he do that actually makes him prize-worthy? Now we're going to talk about findings. So this is F number 2. So the findings, let's see if I can do this now. Ooh, I'm not sure. Let's see. So the findings that Fred made are these. So many years ago, when I was about 6 years old, Fred was a postdoc. So he was a little older, probably not older now, but he was older then. And Fred discovered something that was heretical. And that's part of the reason he's our friend. So it was heretical to believe that the genomes of cells could alter. Okay? So it was a given that if you had a genome in one of your cells, that those cells had that genome until you died. Okay? Fred threw all that in the trash. And so what he found was that if you had cells which had a stable genome and you gave them a chemotherapeutic agent, their genomes went to hell. Okay? And the way they went to hell was by amplifying a particular gene, in this case, one that was the target of that chemotherapeutic, which said that cells are capable of doing all kinds of things with their genomes that none of us ever thought was possible. This was a eureka moment. Okay? Now, Fred, if you know him, know that he's pretty driven, and he's pretty tough, and he doesn't stop with a success. So what did he do? He said, well, okay, so if cells can have different genomes, how come they don't all have different genomes, right? And so his second major finding, so that finding was enough. That would have been enough. Never mind. The next one. So the next one is if cells can have these genomes that rearrange, why don't they? And so Fred discovered this whole new thing about how chromosomes recognized machinery that kept them from rearranging themselves. And cancer cells didn't use that machinery. So that said that normal cells not only wouldn't do this unless there was some insult or another, but they wouldn't do it if they had this machinery activated. And of course, cancer cells don't have the machinery activated, and they rearrange themselves a lot. So that was the second thing, and that's called non-homologous end joining for those of you who are aficionados. So 2 major things. 2. Yeah? That's F number 3, findings. Yeah? So findings. So we're now at number 4. So what's number 4 for Fred? Number 4 is that Fred loves to go fishing. In fact, if you go to the harbor in Boston and you ask a fisherman about Fred Alt, they'll say, you know, Fred is a great scientist. And if you go to the Children's Hospital or the Dana-Farber, they say, but Fred is a great scientist, a great fisherman. So he's either a great fisherman or he's a great scientist. Depends on your point of view. Actually, from my point of view, it's both. So Fred is a great scientist, a great fisherman, and a great friend. And I think all of those Fs certainly fit F. Fred Alt. And so I'd like to just introduce Fred Alt to you on behalf of the selection committee, on behalf of the NFCR, and on behalf of all the people who exist here as the next winner of the Albert Szent-Györgyi Award, and I am absolutely honored to welcome him into that small brotherhood, hopefully soon to be sisterhood. Fred?
Well, Webb, thank you very much. I mean, you said what I did a lot better than I could and probably gave me a lot more credit than I deserve, but thank you anyway. And it's also really nice to follow Senator Markey up here. I think that talk was exceptionally inspiring. It was the kind of thing that a lot of us sitting around here need to hear. So I just hope it keeps happening and he keeps pushing forward with it. So I'm really honored to receive the Albert Szent-Györgyi Prize from the National Foundation for Cancer Research. Very humbled to be included with the past winners. I had known all of them, or almost all of them, but I've always admired all of them for their incredible contributions. We know that cancer is a complex and terrible disease. I learned that early on, as Senator Markey said. Both my parents died from cancer, actually in rapid succession, when I was in elementary school. And actually now, yes, lucky to have the slides there. And I think on the lower side there is my father and mother in earlier years, and later is a picture of my mother and I think pretty much just before she died. And another is a picture, it's not me, that's my brother here, who everybody out here says, is that your father? You know, and my father just before he died, but yeah, there he is. So he was 14 years older. But at that point, it did make a big impact, and I wanted to become a cancer researcher, but doing that and then making an impact, et cetera, those are all different things. Took a lot of fortunate circumstances. So I was a cancer orphan, but I was lucky enough to be raised by my sister, and I can do that next slide. And lucky enough to even be with my sister with quite a large family that grew to be 6 other kids in the family. But so that really, those were things that helped out a lot in that early part of life. And coming out of Western Pennsylvania in a coal and steel town, I was also really lucky that I was able to go to Brandeis University, as Senator Markey mentioned. Brandeis in the late 60s was a big culture shock. It was an interesting place to be, but I think the thing that really set it apart, and still does, it was a wonderful research environment. And I was able to work in a lab. It was a plant biology lab, but I was able to work in there with a great mentor for all 4 years, and I learned about what it was like to be in a lab, going to lab meetings, spending all your time in the lab, getting takeout Chinese food, and eating all that stuff that we all know. And it was great, and it gave me the basic training. And plant biology was perfect training for doing cancer research. You learned about metabolic pathways, you learned how to do experiments, and it's what you needed to learn how to do. I went on to Stanford, and when I went to Stanford, I had hoped to study gene regulation in cancer cells, but there weren't that many people doing it. I took a pharmacology course from the late Robert Shimke, who just died this year. At that time, he was a young professor, and everybody knew him. We were talking about him last night, some of us, as being a very smart, tough, creative, and somewhat eccentric guy, but he was a perfect person to work for. And when his lecture, I listened to him talk about this mysterious phenomenon of cancer drug resistance. That sounds like the right kind of thing. And I asked him if I could join his lab, and he said, well, why don't you give me a proposal? So I heard what he was talking about, so I proposed it, and he thought that sounded pretty good. And so he invited me to join his lab, but he didn't give me a slide. He said I should spend a little time. He wanted to see what I would be like in real life. So that was an interesting time, 1971. Scientific strategy at that time, one of the real things that people did, and sometimes still do, I know Hita over here likes that kind of work. You had to purify a protein and then once you purified the protein and you wanted to study it, you had to make antibodies against it. And that was, in those days, not as simple now. Now we get it from a catalog, but back then it was different. And that was Stanford. If anybody knows about Stanford, its nickname is the farm. And that was what it was. We had to purify the protein, then we had to take it out behind the museum where there were big pens, and we'd inject it into goats, sheep, rabbits, and we'd make our own antibodies. And the goats and the sheep were in a pen near the lab, and there actually was a big cow in there as well. And the tough part was actually catching them. So, yeah. And yeah. So I think this, whoa, went too far. So this slide, I think the one up against the wall is actually Bob Shimke at that time. I might be the one behind him. It's hard to tell with the hair back then, but it's, so I'd played a little bit of football when I was growing up, and we had to catch these goats and sheep. They would jump over our heads. They'd run through us. Sometimes teeth were lost. Science was tough. But I made a few leaping catches, similar like Bob's, and he thought that was fantastic. So he said, "You can come work here," because he knew I'd be good. He knew I'd be good for something, and so at that point, we decided to study how cancers become resistant to methotrexate. As Webb mentioned, it's a drug that inhibits an enzyme called dihydrofolate reductase, and it's also known as DHFR for short. And that's a crucial enzyme for cells to grow and divide, and cancer cells grow and divide rapidly, and when you treat them with this drug, it kills them. It's actually was and still is very successful for treating a number of different types of cancers and other diseases. But the thing that is the problem is once the cells you start treating with the drug, pretty soon new cells grow out and they become resistant to the drug. And then you give more drug, and then you give more and more drug, and the cells just keep becoming more and more resistant, and eventually you just can't do anything more. So you have drug resistance, which is a big problem in cancer. It still is. So we wanted to figure out how they become resistant to the drug. And it was already known that they have higher levels of the protein. So we wanted to grow the cells, and at that time, mammalian cells weren't really grown very much in culture. We didn't have any experience, but Bob had some faith, and he actually got the NIH money at that time to go buy some hoods and incubators, and I bought a cell culture book, and yeah, and then he called up Roswell Park Memorial Hospital, where there was a lot of cancer chemotherapy going on, and he got the cancer cells that we needed to work with, and then we got pills from Lederle where we could actually grind them up and purify the methotrexate from them, or it was called aminopterin. And you'd select the cells and then we had to purify the protein and make the antibodies, and found out really there was more enzyme, and that's when the goat and rabbit skills came in really handy. And that worked out pretty well, and I got cleared to finish my Ph.D., and I had it all lined up to go work with David Baltimore, who was a Nobel Prize winner at MIT. But this whole idea, but how do they make more enzyme? That was really, so I decided to stay an extra couple of years, which turned out to be a really good thing to do. And as Webb said, the conventional wisdom at that time is that we have lots of genes, but you get one gene from your father for each protein, one from your mother, and that's what you have. And for a variety of reasons, we had this somewhat, I guess at the time, heretical notion that cancer cells might be able to increase their numbers of gene copies. So we wanted to do that, but then there was a problem at that time because it was really early on. How would you get a probe for a gene, and how would you do that? And I won't bore you with that. It was a long shot we did. We had to try to pull a needle out of a haystack. We got a little bit of radioactive DNA for the dihydrofolate reductase gene, and then took the cells and we analyzed them, and the curve's not that important, but it's a log scale, if you can tell, so things go up by powers of 10, and the cells that are just the normal parental cells are on the right-hand side of that thing, and up here, yeah, this side, my left, and you can look at the middle points of the curves, and what you found is the cells that were very resistant had hundreds or thousands of copies of the gene, so that was really a shocking result, and the curve in the middle is what happens when we took the cells out of the drug. They actually lost the gene copies. So it really showed that mammalian cancer cells could have unstable genomes. They could gain genes when they were put under selection for drug resistance, and they could lose them when it was taken away. So that was, that was, for us, that was a very fascinating result at the time, and led me right where I really wanted to be, is right in the heart of cancer research. So I finished there, and then I went to work with another amazing scientist, David Baltimore, and we worked on gene rearrangements used by lymphocytes to make antibodies against pathogens. And again, I won't go into bore you with the details of that work, but in trying to figure out how lymphocytes did that, we know there was a mechanism by which cells would break and join chromosomes. So we tried to figure out the joining mechanism, and in doing that, found this pathway that Webb mentioned, non-homologous end joining, and then down the road, work of others, including Andre Nussensweg sitting there, and a number of us, we tried to figure out what non-homologous end joining does in general, and we found out that it's very, very important to keep genes from rearranging and causing translocations in cancer cells, and causing amplifications in lymphomas and other cancers. So it wasn't exactly what we were looking for, but it turned out, again, to be very relevant. We also started amplification studies in the Baltimore lab. That's because a woman at Sloan Kettering, June Beidler, had found out that, and again, this will be a hard one for me to show you, but I mean, just to through my model here, you start out with a cell, a chromosome, a gene, and you make an enzyme from it. And then when you inhibit the enzyme, what happens is you get lots of gene copies, and you get more enzyme, but the more drug you put in, you just keep making more and more enzyme, and that's what's left over, and the cells continue to be resistant. And then where are the gene copies in cells? Well, what's up here is one side, you find the normal chromosome on the top, and then you find chromosomes that have long regions in them that look really strange, and that's because they have single copies of genes amplified. And in other cells that are unstable, down at the bottom, they have lots of gene copies just scattered all through the nucleus of the cell. We knew from our methotrexate resistance that is what amplified genes looked at when you looked under a microscope at a cell. So we knew that was evidence they were there. She had found these, but she'd found them, like the ones you see there, in human neuroblastomas. So all of a sudden, human neuroblastomas, not even tumors that had been undergoing any drug treatment or anything like that, but they had these evidences of amplified genes. So the question was then, what were the genes that were amplified? And so we started trying to figure that out already when we were in David Baltimore's lab. And the idea we had was that there would be a gene that actually caused the cancer, not one that would cause drug resistance, but one that made the cancer grow better. And there was reason to think that because Peter Vogt, who was back there, who won the Szent-Györgyi Prize, had discovered the first cancer-causing gene in chickens years earlier. So you knew those genes. People wondered whether they'd be in humans, and then while we were trying to do this, a number of human genes were discovered. And I just wanted to say that before I finish that story, when you were in the Baltimore lab, there are always exciting things going on, and I just want to do a little anecdote about what happens in the lab, just to sort of show you how exciting it can be. So I was working in the lab next to a guy named Owen Whitty, who just happened to get the Clues Award for the AACR. He came in really excited one day, and he was showing me this autoradiogram, like we'd get on an X-ray film, and it had a couple little black bands in it, and he was very excited because he found that a viral cancer protein had an activity that modified a particular site on other proteins. And he said, well, so where's that going to lead cancer biology? You know, this little black band on a gel that he's very excited about. But on that basis, this gene ended up being activated in a number of major human cancers, CML for one, and another Szent-Györgyi Prize winner a few years ago, Dr. Alex Matter, designed a drug to inhibit that specific activity that Owen found that day. And that drug, Gleevec, turned out to be the first smart cancer therapeutic drug. It specifically kills a particular kind of cancer based on the gene that's activated. So I have to say that there are more of those now, and I just would tell that anecdote because it was a privilege to be there and watch right at the beginning of something that I think pretty much totally fits the National Foundation for Cancer Research's model of research for a cure. I mean, that's from beginning to end is what you'd like to see. So we finished the neuroblastoma project at Columbia a few years later. My student discovered the amplified cancer gene that was in neuroblastomas. It was related to a viral oncogene and an oncogene that's activated in B cells, and based on that, we named it neuroblastoma MIC. and we found that it was amplified in a lot of neuroblastomas. But then Jeff Trent and other clinical oncologists who were studying the tumors found out it's only amplified in tumors that are much more aggressive and are much further along. And so that was really important because it became a diagnostic for neuroblastomas, and it also changed the way that neuroblastomas were treated. So again, a very aggressive, deadly childhood tumor, but it did make a difference. And again, that was extremely satisfying for us because it showed that just for gene amplification, it was not only a mechanism by which cancer cells become drug resistant, but it was also a mechanism by which they get generated. And again, we found all the basic stuff, but the others translated it, and that ended up making lots of substantial contributions. So just finish up with a couple of things. I have to say that I had many great mentors. That makes a huge difference in one's career. I've also had great trainees. I'm going to name two. One was Ron DePino. Name him among other reasons because he won the Szent-Györgyi Prize a number of years ago, so he beat me certainly to that. And he's done a lot of things, but used his mouse modeling and other skills to find links between cancer and aging. He's also become now the head of the MD Anderson Cancer Center, and we heard the phrase moonshot. Well, it was up here a little. That was the one that he developed that Senator Markey mentioned in terms of where we should be going with cancer research now and what should be on the horizon. I mentioned the other one, since he's here. George Yankopoulos is sitting over there. So George and Ron were in the lab at the same time. He is the leader of Regeneron Pharmaceuticals, and they've developed a number of blockbuster, amazing drugs, and they've got many more coming, I think. But I just mention him because I believe George recently was down here to meet with President Obama, along with some other leaders in the pharmaceutical industry, to discuss precision medicine and to talk about how basic and applied research can cooperate to make better therapeutics. Correct? I think, yes. Okay, so, and again, we have to look at that side of it too. So the last thing I want to mention is friends in science. This one. So last year's winner was Jim Allison, who was an immunologist. And I got to know Jim really well, mainly because I'm also part-time, at least, an immunologist, and we ended up together at a lot of meetings. And when we go to meetings, we do things other than talk science sometimes, and Jim was up being a keynote speaker at our programs, in that case, our institute's retreat, so took him out fishing, and that was the result. So he was pretty happy. But with Jim, I want to say a recurrent theme that I've talked about the whole way through. Jim did basic work in immunology. He was an immunologist. But his brother died of prostate cancer. Jim had prostate cancer, and he was, for a long time in his life, he was very focused on trying to bring his work into cancer research. And his work found out a way that many people had tried, but he figured out how to do. It led him to be able to generate a drug, in this case an antibody, that now works very broadly, but he showed that it extended the survival of very late-stage melanoma patients who just weren't able to be cured, but it cured at least a significant fraction of them. And the key thing about that drug, the immunotherapy type of drugs that I think everybody's excited about now, is it provides a template for a new class of drugs of that sort, and I think all the indications are they're going to be a lot stronger and they're going to be a lot more broadly applicable. But again, it's a great example of how somebody doing really basic research in the lab made a discovery in cancer biology that is kind of just remarkably important as we go forward. So. Like it was when it took my parents more than a half century ago, cancer still is a complex and terrible disease. But I think with each new discovery, all these discoveries, it's going to become less mysterious. It has become less mysterious. It's more understandable. We don't fully understand it. And it's becoming and will become much less deadly as more research goes on. And I think the cancer research field, with the support of insightful organizations like the National Foundation for Cancer Research, is now really set to make great progress. It's progress that's becoming more and more rapid. And you've heard the mention of genomics and now post-genomics, and that's an era that now provides us with an arsenal of new technologies that I would say 2 or 3 years ago we couldn't have even dreamed about what we can do now. And it's actually staggering because it's so much that can be done. And now I think more than ever, and I think Senator Markey said that so incredibly nicely, that now is the time, if we have the opportunity and if we get the support, I'm confident that basic cancer-driven research will help exploit this current technological explosion, and it will get more and more breakthroughs, and it's going to be at an ever-increasing pace. So I'd like to, again, thank everybody here very much. Again, I'm very humbled and honored to get this award, and it's wonderful. The last thing I'll put up, because this is an example of what cancer genomics looks like, something we did, except I'm just showing it because one of my postdocs made it as a poster, and I think it's a nice piece of artwork, and just showing how translocations go throughout the genome. And the last thing I want to say is that I have 2 of my best friends that are here, Flu and Tom Look, and they also are in cancer research and immunology, and we also do a few other things. Thanks.